In this video, we're going to be learning about what we call the beginning of the payoff phase to glycolysis. Now, in order to understand why we call it this, we need to think back to the initial method that we used in order to get glucose to break, which was simply by adding two phosphate molecules in order to increase the amount of negative charge that exists within a glucose molecule, thereby making it more unstable by increasing the potential energy of glucose and allowing it to spontaneously break. Now, the problem about this phase is that it consumed ATP molecules, specifically two ATP molecules, one for each of the phosphates that we added to glucose. Now, obviously, we don't want to spend energy in order to produce energy, but this cannot be avoided because this ATP that we added is effectively the energy that we supply in order to get our chemical reaction to start, meaning that this is our activation energy. And starting now, this is where we actually begin to get some of our ATP back, and it requires an enzyme that we call a dehydrogenase enzyme. Now, from this name, the function of this must be pretty self-explanatory. DE tells us that we're going to be removing something, and what we are removing is obviously a hydrogen, but it's not exactly that simple. So the target of our dehydrogenase reaction here is the molecule that we had two of after the splitting of glucose, which of course is glyceraldehyde 3-phosphate, or G3P for short. Now remember that because glucose broke in half, we now have two of these, so every one of these reactions is occurring twice. So for every product that we get, we actually have two of that product because, again, we have two G3P molecules as our reactants. Now, the reason why G3P dehydrogenase is a relatively complicated enzyme is because we actually get to see a coenzyme that is required in order for this reaction to function. Now, the reason is, if we were just removing an H+, that would actually be relatively simple, but we don't just want the H+, we also want the electrons within the bond that anchors this hydrogen to the carbon on the aldehyde group of G3P. Now, this means that we need a fairly strong positive charge, which actually comes from the coenzyme here, which is what we call NAD+. And for those that want a mouthful of a name, this compound is what we call nicotinamide adenine, again referring to the nucleotide that we, or rather the nitrogenous base that we see here, dinucleotide. So nicotinamide adenine dinucleotide, which does tell us a little bit about the structure in that it has two nucleotides here, one of which being adenine, but most importantly, it's the positive charge here that allows NAD+ to act as what we call an electron carrier, and more accurately, what we call a potential energy carrier, which will be very useful in one of the later stages of cellular respiration. Now, the mechanism here is a little bit complicated and involves a fairly complicated step involving the actual enzyme itself, but the gist of it here is that the two electrons within the bond that anchor hydrogen here actually migrate, being attracted to the positive charge on NAD, which means that this carbon here on the aldehyde group has now lost two electrons and therefore is going to have a very strong positive charge because it is effectively missing a bond. Now, as a consequence of this, this positive charge is going to attract to a strong negative charge, which happens to come from a phosphate ion. Now, this is important because this phosphate is what we call an inorganic phosphate, meaning this phosphate is not actually attached to an ATP molecule, because remember, we want to minimize the use of ATP wherever possible because we don't want to spend energy in order to produce energy. And as it turns out, your cells have a large amount of phosphate that is basically just floating around in the cell, acting as a pH buffer. So remember that phosphate can act as the basic form of a buffer, while phosphate that has a hydrogen on it like this can serve as the other component, or the acidic component of the buffer. But as it turns out, again, this phosphate with its strong negative charge will form a natural attraction to the carbon that is now missing a bond as a consequence of NAD plus and G3P dehydrogenase, producing this molecule that we see here, which again looks like glyceraldehyde 3-phosphate, only now we have a phosphate where this hydrogen used to be, and this is what we call 1,3-bisphosphoglycerate, because this is effectively having a phosphate replace the OH of an acid functional group, and we can abbreviate this as 1,3-BPGA for short. Now, as a brief preview to what comes later, to understand how NAD plus functions as a potential energy carrier, we can take a look at the movement of electrons. So remember that whenever we have a decrease, or generally whenever we have a decrease in the number of bonds that a compound forms, usually we have a drop in potential energy as well, because electrons ultimately determine how reactive a specific chemical is, and when one bond moves from one molecule to another molecule, we have a potential energy transfer. So when this bond here migrates over to NAD+, the potential energy of G3P decreases, while the potential energy of NAD+ increases conversely, because these electrons are now over here and now able to do a chemical reaction that NAD+ was not able to do before. And this transfer of potential energy will be useful specifically in the electron transport chain. But we will wait for that video series to come up. For now, what we're interested in is the molecule that we produced by adding this inorganic phosphate here, because this is obviously an unstable molecule. In fact, let's put an exclamation mark there, because of the incredible amount of charge repulsions here. Now, this here is key. The idea of adding two phosphates to a single molecule to increase instability is one of the keys to allowing cellular respiration to function, because one of these phosphates here is going to be used to make ATP. So let's talk exactly about what method that uses. So this molecule here looks like ATP, only we only see that it has two phosphates rather than three. Therefore, this molecule here must be a DP, adenosine diphosphate, and we can make ATP by simply taking one of these phosphates in order to transfer it to ADP in order to make ATP. Now, the problem about doing this is that ATP is a very difficult molecule to make, because it involves forcing three phosphate ions close together like this, overcoming all of the intramolecular repulsion that normally keeps them apart. So the key to doing this is creating a molecule that is even more unstable than ATP is, with a higher potential energy. So this means that the potential energy of 1,3-BPGA must be greater than the potential energy of ADP, allowing this phosphate here to transfer as a result. And we'll take a look at what exactly is needed to do this. So the enzyme that is required, again, whenever we have a transfer of a phosphate, we're going to involve a kinase enzyme, and in this case the target, of course, is 1,3-BPGA kinase. So we're going to have a phosphate transfer with this molecule as the target. Now, this phosphate here on the bottom, the one that we added from the buffer, is the more unstable of the two, and we can see that because of its location. Because of the fact that it's close to this dipole, or this negative pole on the double bonded oxygen here, whereas this phosphate here is close to an OH group, which does have a negative pole, but there's also a positive pole in order to stabilize this negative charge. If we want to go into more detail, we can see that there's a double bond here, and a double bond is made of 4 electrons. So between these electrons in the double bond and this negative pole on the oxygen here, this phosphate here is easily the more unstable enzyme, or on the 2, which is why the enzyme makes this phosphate its target. So the reaction that the enzyme catalyzes here is the breaking of this bond that anchors the phosphate to the 1,3-BPGA molecule, and then that bond is going to reform on the phosphate, or on the phosphorus atom here on ADP, therefore transferring that phosphate and allowing ATP to form again, because this phosphate is even more unstable than adding it to a second phosphate on ADP would. And this is what we call substrate-level phosphorylation. Now, the name phosphorylation is pretty self-explanatory. That means that we are adding a phosphate onto a specific substance, and by doing this at the substrate level from one reactant directly to another reactant allows us to produce ATP directly by transferring one of the phosphate ions from one of the substrates of glycolysis directly to ADP, and we'll see how this compares with how ATPs are made at later stages. And after this, we can see that our 1,3-BPGA no longer has a phosphate here because that was transferred to ADP, leaving us with a molecule called 3-phosphoglyceric acid, or simply 3-PGA. In the next video, we'll look at the fate of 3-phosphoglyceric acid and how we can use another example of substrate-level phosphorylation in order to transfer this remaining phosphate here to an ADP molecule to produce one more ATP within the process of glycolysis.